Hello, everyone. Thanks for staying around for this long and very exciting symposium, I think, and thanks for David for organizing all this. So I'm going to tell you a little bit about a regional perspective from the Arctic and what we see about arthropod population dynamics in that region. I'd like to start with a little bit about the drivers that we see as most important in the Arctic, because actually I think the Arctic region represents an opportunity for contrasting the global change drivers and their influence on insects and arthropods in general compared to other latitudes. So in the Arctic, we obviously have very little agriculture and very little human land use influencing habitats altogether. So pesticide use is obviously of minor concern, and we have very little issues of habitat fragmentation. On the other hand, as you can see in the panels to the right, we have quite strong climate change already happening and predicted for the 21st century. So the panel to the left gives temperature changes for the northern hemisphere altogether with two different climate scenarios, the most pessimistic one in red, which we're actually tracking right now. And in the panel to the right, you can see how those changes are going to be spelled out for the Arctic region. And clearly that's more pronounced, but it's also an area where temperatures during the growing season are actually hovering around zero. So for insects and other organisms that rely on fluid water, actually an increase of 10% within this decade is going to be a substantial change to populations of insects. So some of this change has already happened, and I think, yeah, the Arctic represents an opportunity for comparing the role of climate change and other land use drivers in that region. So I lead a network of scientists who are interested in arthropods in the tundra region, and we're pretty excited about this map that I'm showing here, which we recently compiled as a kind of status for how the diversity of just one group of arthropods, the spiders, are varying across the sub-regions of the Arctic, the subarctic, low Arctic, and high Arctic region, and across the different continents or regions of the Arctic. So on the one hand, there's some exciting details about how the diversity of particular group of arthropods, the spiders, is made up and how that changed with latitude. But I think the point I want to make with this figure here is that actually we have very little understanding of who is where when it comes to arthropods or insects in particular in the Arctic. So we have some really big pie charts for very large regions of the Arctic, and we've got nowhere near gridded data or detailed compositional data from high-resolution maps. On this same map, we also have a scattered number of small red dots you might be able to see. So there's about 10 dots there, and those are actually the sites in the whole Arctic region that do some sort of arthropod or insect monitoring. And among those 10 dots, I would say only a few of them have a good long-term handle on insect abundance. So I'm going to argue that we need obviously more of that kind of information, but I'm also going to show you a few examples of how changes occurring at those few sites where we have some abundance data, what they look like. So I'm going to take you to this place in northeast Greenland where one of those dots are, a site called ZACKENBERG, which is the home of a rather long-term monitoring program. It's not just a monitoring program for arthropods or insects, but actually a kind of ecosystem-based monitoring program that covers also birds and mammals and the whole abiotic environment as well. But arthropods is one component of that, and of those, obviously insects make a great part. So some years ago, we were able to demonstrate that for some of the families of insects for which we have abundance data, we actually already then saw quite substantial declines in two groups of Diptera, the chironomids and the musket flies. At that time, and as part of this funded long-term monitoring program, we do standardized trapping in the Martin Sorg sense, if you like, not by malaise traps, but by pitfall trapping. And the samples are actually counted the old traditional way, but none of the specimens I identified to the species level. So at that time, we only had kind of abundance data for insect families. So since then, we've done quite a substantial effort to try and identify the specimens of some of these samples. And I'm going to show you some of the results here from the musket flies. We've had a really good PhD student who spent a lot of time identifying more than 20,000 of these specimens. Luckily, Diptera in the Arctic are less diverse than in Costa Rica. And for musket flies, we got a total of 16 species. So that's quite mind-boggling. For musket flies, 16 species. That's all we caught over 18 years of monitoring. I should say that the pitfall traps are yellow, and because of the vegetation about 2 to 3 centimeters high, we can use them basically as pen traps, so they also catch the flying insects quite effectively. So these panels here in the leftmost column show family-wide abundance trends for 3 different habitats: the dry, mesic, and wet, basically wet habitats. And as you can see, panels with straight lines have significant trends, and none of the trends are increases. So we've got in the dry and the wet habitats, we've got significant decreases, and for the wet habitats, these were actually around 90% declines in abundance, so quite strong negative trends. Overall, the abundance trend for musket flies was about 80%, so similar to what we've seen in Germany, and this is without pesticides, I should say. So on the panel to the right, we have broken this up into individual species, and we were kind of looking at whether, like there's been some indication of in the previous talks, whether these broad taxonomic groups would hide in species-level differences in these trends. And actually, if you look on the top row, the dry habitats, we do see an overall trend in that family of musket flies, but when we look at the individual species, some seem to be going down in some years and others in other years, and they kind of cancel each other out. So in the drier habitats for musket flies, we don't see that any of the species are significantly declining. Kind of the opposite is going on in the mesic habitats, where overall we see no decline, but there are certainly some, particularly of the rarer species, that are showing significant declines. And in our wet fen areas, actually the overall very strong decline is also matched by most of the individual species. So by breaking this up into habitats, actually we get a mix of different responses, and also different patterns when we look at broad and fine taxonomic resolution. So although this is quite a substantial monitoring program, and it does do quite a good job at monitoring the abiotic environment, we had actually, to be fair, quite a bit of trouble finding good climate predictors to match to these abundance trends. And mostly we would say that our year predictor, so the trend across the time period, was better than any of the individual climate variables, which I think is kind of interesting to keep in mind, that even for an area where we thought, oh, this has to be linked to climate, we didn't actually get strong support in our statistical models for any of the individual climate drivers. Now, one of the things that are maybe missing from the set of drivers that are measured at that site is information about moisture changes in the topsoil. And that's obviously important for larval habitat for these insects. And we suspect that actually, had we had better information about moisture levels in the topsoil, maybe that would have been the smoking gun for these populations. So we are trying to get a better handle on that now. So we also have some data for spiders, because we were actually wondering a little bit whether these systemic changes in abundance, whether they were actually partly driven by our own effort to sample these populations. So we were a little bit worried about the scientist effect on population sizes. So it was in a way comforting to see that some of the very active arthropods on the ground, that they were not depleted by our efforts to monitor these populations. So for spiders, we have a mere 9 species in the whole data set, monitoring spiders for almost 20 years. So that's also quite striking, but it gives interesting opportunities for looking at individual species population dynamics. Also here, we didn't find any species that were in increasing abundance over the course of this 18-year period. We do find some more specialized lineaphiid spiders, so small orb-weaving spiders, that were in decline actually rather strongly. If you look at the individual years, I think maybe we've got something to learn about how interannual variation can tell us a little bit more about the drivers behind the change in abundance. And particularly for our spider data, we were seeing quite strong density-dependent effects, so kind of importance of abundance in previous years, and maybe that's something we can learn more about. So for spiders, it's well known that they eat each other, so maybe that's what we're seeing with the spider data. Okay. So we also looked a little bit at the whole community and how that community would change through time, not at the species level, but in broad functional groups or also broad taxonomic groups. And these two panels show some community compositional changes in the form of non-metric ordination analysis. And what we try to show with these figures is that herbivores and parasitoids generally become more abundant at the expense of decomposers such as Collembola, and we see that there's a habitat-specific magnitude of those changes, and particularly we see changes in the drier habitats from this one site. And I think there's been some talk about extreme events or at least variability. What we see driving the changes in community composition is partly the variability in the shoulder seasons. So that's the freeze-thaw cycle dynamics of the study area. So that's interesting too, I think. Okay, so we've been counting a lot, as you can see, and I think I'm going to end this presentation by showing you a little bit of a glimpse into how we would like to count less in the future and get more data nevertheless. So we're trying to use different image-based methods to help acquire more and higher resolution data on both insects in the field and the samples that we collect and work with in the lab. So in the video to the left, you can probably see some flowers, and maybe if you're lucky, you see some flickering insects that are visiting these flowers as the day progresses. Those are syrphid flies visiting a very common Arctic flower species. And in this way, we can actually use computer vision tools to extract information about the insects that visit the flowers. In the lab, we can identify species automatically using image-based methods. So there's going to be a couple of talks about this tomorrow in the afternoon. You can go and check those out. I can't get into more detail right now. So I'd like to end this by saying that in the Arctic, we're assuming that the insect variability is really driven by climate change, and that these responses are uneven among species, functional groups, and also habitats. And we suspect the drying of topsoils is really going to be important as the permafrost thaws. But we need data from more sites and also with more detailed monitoring of the drivers. And I think these new tools could help us in the future. It's not just in the Arctic where the species diversity is low. So I'd like to thank David for putting this symposium together, all the co-authors on the papers that I presented some results from, all of those who helped out in the field in the lab. And I'd like to give a word for this network for arthropods of the tundra. We call it the NEAT network, and if you're interested, you can follow on the Twitter handle below. And thanks to you for listening.
Hey, really cool stuff. Do you have any data on aquatic insects that could maybe help you tease apart that idea about the soil moisture being important? Because you'd expect it if you have aquatic invertebrates in the same area, they wouldn't be affected if that were the driving force.
Well, we do catch some of the insects that have aquatic life stage, immature life stages, like mosquitoes, but we don't actually have a monitoring program for aquatic insects altogether. I think that's a good point to tease about those effects by looking at aquatic insects. Efforts in the southern hemisphere to, sorry, I didn't get the question.
Any efforts in Antarctica at the South Pole, and can you make comparisons if possible?
As far as I know, I mean, there's quite a lot of efforts in Antarctica to look at insects, mostly from an invasion perspective. There, as far as I know, there's not really a monitoring program. There are also very few insect species, like 2 on the Antarctic mainland, I think.
I was wondering how this might serve as like a metric for reducing or comparing the effects of pesticide use. So with your data, you mentioned a couple times that this is in the context without pesticides. So how would we maybe replicate or index the same way in agricultural regions to have another new long-term data set that can then compare changes in the Arctic and growing regions and relate it to pesticide use?
Well, yeah, that's a good question. I guess there is also some challenges associated with that. I think there's been some suggestion that we can use gradient approaches, that we kind of have monitoring programs that span gradients of stressor influence. So that would be really large-scale gradients. I think it's also, as we move forward and try to understand the local drivers, I think we can at least work with systems with and without additional drivers to separate the relative influence of these different drivers. Mm.